Today you have the pleasure of listening in on a brief but very informative session on the AOAC official methods of analysis process. So for some background, Lukas has been in the food and dietary supplement testing space for over 15 years, and he works on the team that published the AOAC First Action Approved Potency Method in hemp. And this method was developed in collaboration with Eurofin's Food Integrity Innovation and the University of Chemistry and Technology in Prague. So Lukas, thanks for joining. Let's go ahead and get started. Can you tell us what the significance of the AOAC official method of analysis is?
Sure. So the final outcome of the AOAC OMA program are standardized methods. So probably the most significant benefit of such assays is the high confidence end users can have in the method performance and the validity of the generated results. Stringent acceptance criteria for validation data and thorough scientific review by subject matter experts ensure these procedures are credible and defensible. A major advantage from the testing lab perspective is the fact that approved methods are typically easy to adopt and implement.
Great, thank you. It's definitely important. And can you describe the steps that go into getting a method to be an AOAC OMA?
Yeah, at the beginning of the process, there's typically an AOAC call for methods to address a specific analytical problem. Afterwards, AOAC working group develops a standard method performance requirements document, aka SMPR, that summarizes criteria that need to be met by methods. And then candidate methods and associated validation data are submitted and subsequently reviewed by expert review panel, which is composed of experts in the particular field. If in favor, the reviewers may make a motion to adopt the method, and a vote takes place to grant first action official method status. To move the final action official status, authors need to demonstrate the method is reproducible, which is usually accomplished in an inter-laboratory validation study.
Great. It definitely sounds like it takes a lot of expertise and resources to get this method to that point. So along with that, about how long does this entire process take?
The time needed for method validation is dependent on complexity of the workflow and other factors, such as analyte and matrix scope of the particular method. For multi-analyte methods covering large number of compounds, the data processing and compilation of the validation data is typically the most time-demanding step. The length of the whole AOAC OMA process may also largely differ between methods and can be quite lengthy, especially when additional data need to be generated to address concerns and issues raised by the expert review panel. As an example, with our cannabinoid assay, it took approximately 2 months to develop the method, a month to complete single laboratory validation, and a year to have it approved as a first action official method. I would say that 1 year from call for methods is the typical time to get a well-validated method to first action status.
Wow. Well, thank you so much for sharing your expertise and your insight. Certainly informational and valuable information to know. I appreciate it.
Thank you, Vanessa.
Bye-bye.
Bye.